Okay, guys, looks like we're live for our second stream of the night. This will be 2 of 3, so stick around. We got one more after this. That'll be more of an open discussion. I believe Snake was right. It's going to be joining us there as well with Jeremy. But this one's just going to be a back and forth, easygoing dialogue on Ancestry, the case for Ancestry, universal or separate. Obviously, Snake holds to the universal common ancestry, and I hold to that of biblical creation, for example. So we've had many debates and discussions in the past. That's why we're just going to keep this a little bit more of an organic, free-flowing dialogue. Snake is my guest here tonight, so I'm going to let him kick it off, say what he's got to say, and kind of just go from there, and then I'll respond to what he said. But we're going to try and keep it on topic as much as possible. But if we venture off, that's fine too. It's just more of a, like I said, organic, free-flowing discussion. So, Snake, thanks for joining me tonight. Looking forward to this. Yeah, how's it going, man? Kick it off.
Yeah, well, I mean, basically, there's assuming abiogenesis or, I mean, even any kind of biogenesis. I guess there could be multiple times that life evolved early on, but then at some point, pretty much everything that's alive came from this soup of cells that were sharing genetic information with each other. So it's basically last common, not that necessarily everything is related, but last common ancestor would have been a very early life form, a very simple life form. But kind of like the reason I get interested in creationist discussions and things like that is like I just start with universal like animal ancestry, and because it's pretty obvious to me and a lot of people, and then we could even go as far as to say, okay, we can even for now separate plants and animals, but maybe we can agree on some animals have universal ancestry, and then maybe all animals do, maybe all plants are descended from one plant, and then from there on, like I think the more controversial topic is like, okay, so then how did animals and plants come from single cells? But again, you guys aren't going to agree that all animals are related anyway. But that's what I'm interested in mostly, because it's pretty easy to see the patterns of, like, for example, mammalian ears. Reptiles don't have the ear bones that mammals do, but we have fossils that show the reptile jaw bones becoming smaller and smaller and moving towards the ear, and then all of a sudden the ear bones occur basically in the fossils that look like they're half mammal, half reptile, and all of a sudden the mammals have a solid jawbone while reptiles have fragmented jawbones, and those analogous fragments are now in the ears of the mammals. And then further back, we have examples of animals in the lower layers where it looks like they're starting to have ribs that cluster more towards the mouth. And that's kind of why, like, fish and reptiles have multiple bones in their jaws instead of one bone, which, like, from the movement of the ribs into jaw bones. So it seems to me that if these animals were created in their current form, there's no reason to leave all these breadcrumbs. Like, why wouldn't the reptiles just have a solid jawbone? And why wouldn't they also have ear bones like the mammals do? And why would God need to design mammals with a solid jawbone but not reptiles? Things like that. And I did have a couple of questions that I wanted to ask you too, if we could just get to those also. I don't know if you wanted to make a comment on that or just get straight into the questions.
No, I appreciate what you said there. Yeah, we can definitely get right into the question. I'll respond real quick, I guess, to the bigger picture of everything you said, because I think it's important if we're going to discuss whether or not all life is related or can be traced back to one or a few single common ancestors, or if separate ancestry is, I guess, better explained by the data, as we would suggest based on a literal interpretation of Genesis and biblical kinds, for example. So I think the most important way to approach this topic is to focus on that which is differentiating evidence in this debate, right? For example, if I were to look at the sky and say, hey, the sky is blue, therefore biblical creation is true. Well, that wouldn't make any sense since both models can explain the data. And I believe the patterns that you're referring to, patterns in morphology, anatomy, ear structure, for example, patterns in genetics. We can look at mitochondrial DNA, nuclear DNA, highly conserved mitochondrial DNA proteins. I mean, those patterns exist. I'm not going to deny that fact. That's exactly what we see is nested hierarchical patterns. Now, the question is, do those patterns reflect that of, say, descent with modification, or do they reflect design? Because and I'm sure you've heard me say it before, I would say that these hierarchies, these patterns we see, and whether or not it's DNA, anatomy, physiology, they're also expected and predicted by creationists based on the design model. I mean, even Linnaeus, the father of taxonomy, kingdom, phylum, class, order, family, genus, species, he derived that based on function, for example. So that's what we as biblical creationists would look to. So when it comes to the differentiating evidence, that's what's most important. The evidence that's more so neutral, say homology or transitional forms. Homology, for example, the shared structures we see in the biological world, those can either be explained by common descent in common ancestry, or they can be explained by common design. Fossils are tough to look at because fossils can't always establish genetic relationships, because similarities in fossils that you look at, fossil organisms, say you're looking at morphology or anatomy, for the most part, you don't have any genetics of these fossils. They may not even be due to common ancestry, and the evolutionists would admit this as well. And that's why convergent evolution is oftentimes invoked. And it's tough to even give a clear definition. Like I asked for a definition for species when it comes to fossils to RJ. And that's what he looked to, was morphology and anatomy. So without genetics, it's tough, because to answer that question of ancestry, you have to look at that which is inherited sperm and egg. Right? A rock, a fossil, those are not inherited sperm and egg. Genes, traits, and genetics are. So that's why when it comes to breaking the phylogeny or determining what is true, universal or limited ancestry, things that can differentiate between the two models would be molecular clocks and DNA function. So that's certainly what I would focus on here. You did look to like the mammal, like reptiles, for example. You can look at Archaeopteryx. You can look at Tiktaalik. You can look at some living species today that seem to reflect mosaics. For example, the platypus, some of your walking, your mud skippers, for example. Like those mammal-like reptiles certainly are interesting. You spent some time talking about that. They were a highly diverse, highly varied, widely distributed group of reptiles. And like you said, they did have a number of characteristics that are found in mammals today. Now, there are some discrepancies, of course, in the fossil record when it comes to these so-called transitionals. Oftentimes, they suffer from what's called the grandfather paradox. And even when it comes to the reptile-like mammals, more mammal-like fossils oftentimes do occur in the fossil record earlier than fossils less mammal-like. You also see oftentimes geographical mismatches. I mean, the fossil record is just up to so much interpretation. It's tough. And mosaics, once again, I would say that is agnostic to the discussion anyways, because, and you've heard me point it out many times, we would expect the same thing, because if God created us in his image, okay, let's look at the way we design things. and therefore we can get a sense for the way God may have designed the biological world. And it's not just a coincidence. We don't do it on purpose, but we do build in these types of nested hierarchical patterns, and we do build these types of, transitional like vehicles. For example, a military amphibious military assault vehicle that, say, blends the features of both a land vehicle and a vehicle built for the ocean. Even these crossover SUVs, for example, I mean, they blend the features of an SUV and a van, and they're even hard to categorize. So evolutionists would look at those and say, hey, there's your perfect example of transitional form. But these are all agnostic. It's kind of like me pointing to the sky and saying, hey, the sky is blue, therefore biblical creation is true. So definitely my focus would be DNA function, molecular clocks. And like you said, are all plants and animals related through common ancestry? We can talk whether or not pine trees and whales are related through common ancestry, but I like to go closest to home, especially because my starting point is the Bible, and it says that God created two humans, Adam and Eve, that are made in his image, and they're distinct from the rest of the biological world. Therefore, are humans related to any other form of life, I think, is the most important question. Therefore, the most similar creature in the biological world to humans would be chimpanzees, other primates, the old world monkeys, the new world monkeys. We do see nested patterns there as well. So I don't mind going right as close to home as possible and looking at that evidence and seeing if there is a barrier or a breakdown or a separation there in terms of ancestry. So I think for the most part, I can't remember everything you said there, but for the most part, I think I touched on it. The DNA function, molecular clocks, I believe, is what demonstrates a limited ancestry, but we can touch on that in a bit. If you want to, you get all the time in the world as well. You can respond to anything that I said there, Snake, and then we can go on to your questions if you'd like. So go ahead, Snake.
Sure. I think this will lead into my first question. The thing about the transitionals is that it's not just that we're finding a mix of features, but that it's a very specific mix of features. So I'll give a couple examples, but they're exactly where the evolutionary model predicts they would be, assuming that the fossil layers are deposited, chronologically, and then things are evolving between them. And that's important because we're able to, if the assumptions of how these organisms are changing over time is correct, then we should see this particular change in this particular area. And then we go in and we find something very similar to what we were looking for that has been predicted. And so it's not necessarily just looking at it and trying to fit it into something where it looks like it's in between. And it's, I don't really think it reflects design because of what I see as inconsistent design, like what I was saying about the fragmented jaws of reptiles, the fact that why would God need to create multiple segments for a reptile jaw when we have proof that he can create a very similar animal without these segments in the jaws and with a solid jaw? And why wouldn't he give reptiles a mammalian ear and things like that? And the example I like to use a lot also is the penguin. I'm not sure exactly how related you think the penguin is to other bird species, but I guess real quick, do you think that the penguin, like all the penguins were created as a kind, or do you think that the penguins were evolved from some flying bird?
I would say, since the Bible does say kinds of birds, I do believe that at creation, for example, God would have created animal kinds in bulk, in populations, for example. In terms of the ark, God could have brought, like as a working hypothesis, God could have brought anywhere between, 40 and 60 different kinds of birds. And if I believe penguins would be included in the stats and the data that we see in terms of how many bird species exist on the planet today. and it's anywhere between 10 and 12,000 bird species. So, I mean, but whether God created a single penguin kind, a population of them, and only brought a couple of that specific kind on the ark, along with 30 to 50 other bird kinds onto the ark, those could easily speciate into the 10 to 12,000 bird species we see today. And you don't have to answer this question, but my concern is always, okay, if we only see 10 to 12,000 bird species today, but yet the evolutionists will say that birds evolved from a theropod-like dinosaur, a population of theropod-like dinosaurs 65 million years ago, or even longer than that. Why are there so few bird species? And I imagine they would just look to extinction events, for example. But the amount of species, when you actually break it down, if you look at the lizard species, the bat species, the mammal species, cat species, I mean, there really is only a few. And with bird species as well, I mean, this can all easily be explained due to constant speciation rates within 4,500 years since the flood. But an exact answer to how many kinds of penguins God created, or have they the penguins on the ark, I don't believe would reflect the penguins we see today. There's been change, there's been speciation events, but I like to look at the bigger picture. I like to look at it as a whole. Let's look how many bird species, and I believe the penguins are calculated in that number. There's really not too many. One question too, and then you can comment on that, because you mentioned how did those two bones from the reptilian jaw migrate to the mammalian ear in that hypothetical transition? And then I won't jump in on that. Just if you can answer, I'm just kind of curious because you mentioned that, and then you can respond to what I said. Snake, go ahead.
Yeah, if you look at reptilian jaws, they kind of have a bunch of fragments, especially towards like the jaw joint, and so it's not exactly out of the question, even within like creationist genetics, that the size and orientation and proportion of these bones would change. So you have like a couple of like 3 or 4 different pieces like towards the hinge of the jaw, and so maybe those get smaller, and the one that's carrying the teeth gets a little larger over time, and that's just change of traits that is already within the genetic population. And basically at some point they would get so small as they wouldn't be functional for the actual hinge motion, but then they could start disassociating from the jaw and start picking up vibrations. And that, again, that doesn't break any creationist genetics at all, at least as far as changing available traits that are already available in the population goes.
I guess my point real quick is that the number of structural changes and the number of genetic rearrangements that would need to take place, I understand that there could be like a story as to how that's possible. But when you're looking at the actual fossils themselves, you're looking at a fossil, and you're looking at a fossil record that has a tremendous amount of variation and diversity present. And even Aaron Ra himself, in his phylogeny series, he even admits that when you look at, he points to dogs as a basic example, but even he agrees that it's genetics that's the most important field to look to in determining what is related and what's not. Because even in dogs make it 100% clear that anatomy, morphology will oftentimes fail us when looking to determine ancestry, because, and you'd probably agree with this as well, most of the time there's even more diversity within a species than between a species. So that makes it difficult to classify animals only based on their physical traits. So yeah, you can look at this variation and then the fossil record, and you can line things up and make it appear like there was this type of transition from a reptile-like mammal to a mammal. But the fact is, the fossil record does suffer from what's called, like I said, the grandfather paradox. Because you said that it's based on predictions. They expect this. But with the grandfather paradox, for example, and I understand what the explanation is to it, but it is a fact that they oftentimes, let's look at Tiktaalik, for example. They predicted where Tiktaalik might be found, but then it just turned out that there were tetrapod trackways that predate Tiktaalik by nearly, according to the evolutionary paradigm, by nearly 20 million years. Therefore, that wasn't exactly the perfect outcome or perfect prediction. Yeah, they decided to, fit it within the model, for example. But there's, even as I pointed out with the reptile-like mammals, there are more mammal-like fossils occurring in the fossil record earlier than fossils less mammal like. Plus you got geographical mismatches and things like that. So there's just so much interpretation. I mean, fossils can just sing any song that you want. It's genetics. That's why, and we'll get into it at some point, but that's why I believe that molecular clocks, DNA function, for example, do these patterns reflect function and purpose, or do they reflect descent with modification? So go ahead there, Snake.
Yeah, but I mean, the model actually predicts that. It didn't. No one is saying that Tiktaalik was the first and only fish that would have crawled out. Like, there are fish today that are swimming fish, but they're able to go up on land. And we're not describing them as having legs even yet, but we know that fish are capable of going up on land. And so there would be tons of different fish species trying this, and some would be better than others, and some would leave traces. And so far we have traces of probably what looks like a huge range of that happening. And then like what you're saying about the fossil diversity is there is a huge diversity within the reptiles. There's reptiles with huge chunks of their jaw are all like all the fragments are large, and then there's reptiles with relatively small chunks. Some of them are large, and maybe just the most posterior one is very small. And then you have mammals and mammal-like reptiles that they still have parts of that parts of the jaw that are a little bit larger. They're long and thin, but then they have a few parts that are much smaller in the back, and they line up chronologically as well. We're not just putting them in order of how we want it to look. It is in the order that would be expected if things are buried one after another. And the fact that there's this morphological crossover shows us that it's possible for an animal population to actually get smaller and smaller bones. And as we know, also, a single trait can have multiple different functions at the same time. So a bone could pick up vibrations, and any bone can do that now. Like, blind creatures probably use like a seismic sense to feel vibrations around them. It's just that much smaller ones could pick up much finer vibrations because it takes less force to move them. And I guess mostly what we're seeing in the fossil record is they're split lineages. We're not necessarily saying that they're direct descendants, but they're splitting off from common ancestors. And what we're looking at is morphological similarity, and morphology is inherited by genetics. So, this is what convinces me personally, mostly, is that I understand that the jawbones can get smaller, like no matter what scenario we're talking about, whether God created it or not.
But even though, of course, morphology and anatomy is inherited as well, but the fact that there oftentimes is more diversity and variation within a species than across or between species, that means that interpreting the fossil record and drawing lines and declaring relationships can oftentimes fail us. And as I pointed out, even Aaron Ron is in his series points that out. That's why genetics really is the #1 tool that we need to use to determine ancestry, because even with the fossil record, convergent evolution and loss of traits evolution is what they invoke with the snakes. And even when it comes to reptiles, I believe there's less than 5,000 species of reptiles in the world. So that, once again, is consistent with the bird species, the fact that there's only between 10 and 12,000. It just makes no sense. I mean, there must have been a significant number of extinction events in the past for deep time evolution to be able to explain the number of species that we even see today on the planet. And even if you look today at some of these mosaics, I mean, I find the duck-billed platypus to be a very interesting creation. It's got webbed feet, it lays eggs, it's got a duck bill, and it's got traits that could be classified or looked at as reptilian. So you're looking at this animal that's got characteristics of mammals, reptiles, birds. Heck, if it was found in the fossil record and it was an extinct animal, some taxonomists might even conclude that it's some type of primitive mammal. And if every single dog breed today was buried in a flood and these bones and fossils were dug up thousands of years from now, I mean, all types of connections could be made, all types of interpretations could be made. But like I said, I mean, without the genetics, which really is your only direct, these lines of evidence, these observations are nice, but they really are indirect lines of evidence. When you actually look at the direct evidence, for example, molecular clocks, like, what are your thoughts on I mean, I know you've heard me speak of it before. If we go as close to home as possible, for example, let's look at humans and we look at observed mitochondrial DNA mutation rates, just looking at the observed mutation rate, not making up hypothetical mutation rates in the past and fitting them with, calibrating them with the fossil record. Because the point in question is the deep time, because everything you're looking at and the so-called transitions you're looking at in the fossil record, that assumes deep time evolution. But I'm saying that the number of species today on the planet don't reflect deep time evolution. Therefore, that's the point in question. So we just want to look at the mutation rates today, not only in the mitochondrial DNA, but also in the Y chromosome. Why do those only take humans back, both in the mitochondrial DNA and the Y chromosome? Because this is what I find to be fascinating evidence. Why do those numbers, the number of DNA differences, let's just start with the mitochondrial DNA, why does that only take us back to a mitochondrial Eve roughly 6,000 years ago? Go ahead, Snake, take your time.
Well, estimates range from like 200,000 to like 7,000.
Well, the 200,
and that's just going back. Sorry. That's just going back to a common ancestor. So we can't really use those clocks to determine the actual age of them, just like when they split.
Well, I'm not referring to like your 200,000 year dates for the mitochondrial Eve that are derived. That's because they take the dates from the secular papers. So the observed, like, for example, the Parsons paper, that was adopted by the FBI. It's so good, it's so consistent, which takes Eve back 6,000 years. But when you read the details of the paper, they assume evolution. So they'll calibrate it with the fossil record and conclude that Eve, the mother of all living, the last mitochondrial DNA common ancestor, was 200,000 years ago. But remember, that's based on the fossil record. That's based on geology and phylogeny based assumptions. But remember, here, if we're looking to the models and we're looking at the differentiating evidence, I would be questioning the deep time paradigm, the deep time assumption. So I'm not going to assume the deep time evolutionary model when looking at these observed mutation rates. So when we actually look at the actual empirically observed mutation rate for, let's say, mitochondrial DNA, well, that always takes her back just thousands of years ago. So yeah, I understand what you're saying about the 200,000 years ago, but that's only when they calibrated with the fossil record. So just stick to like your pedigree-based studies. You know, why is that not compelling to you? And take your time, Snake, whatever you have to say.
Well, I did also want to get you to send me those studies so I could read it, because I can't quite find what you're talking about. But
of course, I could screen share them right now, but I mean we can't read them on the spot, so it might just be better for me to send them to you, and you could have you read the Parsons paper at least?
I don't think so.
Okay, and that's fine.
But I mean, there's a lot of things. Mitochondria. Well, there's a different differentiation in mutation rate as well.
Right.
We know that sometimes mutation rates are high, sometimes they're lower. That's about all I can say.
They're consistent for the most part, right? And there's certain things like heteroplasmy, and there's certain things that need the population histories, population sizes that need to be considered. But that's why I always say that the deciding factor as to which model is strongest is going to come down to who's making better testable predictions. So I understand with what you're saying that if you assume deep time evolution, you have to calibrate those observed numbers from pedigree-based studies with the fossil record, okay, to come up with the evolutionary Eve date. But like I said before, you know, that's the point in question. So evolutionists can say, hey, listen, in the past, mutation rates in the mitochondrial DNA were significantly slower, because some of them actually say like between 10 and 30 times slower. But here's the thing. Who's making the best testable predictions? And then you're not new to this, Snake. You know that Dr. Jensen has taken the observed mutation rates in mitochondrial DNA in non-African people groups. And he's saying, okay, this is how confident I am with the Eve date of 6,000 years. He's looked at some African tribes where their mutation rate has not yet been measured. And he's made a future testable prediction as to how fast or how quickly their DNA changes or mutates in the mitochondrial DNA. And that's a prediction on print. So in order for an explanation to be scientific and not just post hoc, ad hoc, or a rescue device or non-science, for example, testable predictions have to flow. They have to flow from that explanations. Therefore, I mean, are you aware of any evolutionists that are making mitochondrial DNA predictions on people groups? Let's say some of these African tribes like the Khoisan peoples whose mutation rate has not yet been measured.
Well, I do need to read those papers from Jensen. Like I've read some of his papers, like the synergistic epistasis I found, a couple other ones I found, but I can't find what you're talking about with
I think that's
the mitochondrial stuff.
I think the synergistic is Dr. Sanford in regards to genetic entropy.
Yeah, it was Sanford. Yeah, you're right.
Yeah, and that's fine. And that's fine. We can talk about that as well. But when it comes to molecular clocks, that's why he's saying, hey, you can point me to some fox in the woods, or you can point me to some animal species, and I will tell you, I will predict how quickly that animal mutates. You know, he's going on the offense, and he's making these predictions because we're questioning the fossil record, the deep time claimed evolutionary history, and these just observed mutation rates. And we can even look to the Y chromosome. But real quick on the mitochondrial DNA, I found it funny that, and you can look at these papers before, of course, not only until after the shock of the evolutionary community as to the amount of mitochondrial DNA differences that exist, did they actually look to coalescence to explain all these differences. So they're invoking coalescence as a post hoc evolutionary explanation to explain away the data. But and if you put yourself in the mindset of a young earth creationist, if just basic observed mutation rates in the mitochondrial DNA, just take the Eve date back to 6,000 years, which is exactly what the dates add up to in the Bible, why would we then take those dates and those numbers and calibrate them with the fossil record? Like that's that wouldn't make any sense. No, it makes better sense to now make more testable predictions, which Dr. Nathaniel Jensen has done. And even in his debate with Dr. Vanima, who's an evolutionary biologist, he even kind of praised Jensen for that. He said that's a good prediction to make, and he admitted he can't make it because according to deep time evolution, there's so many different factors, so many different historical demographics, so many different population histories that it's difficult, but it's not impossible. All the evolutionists would have to do is predict how fast, or no, the evolutionists would have to predict, when does the mutation rate slow down? When does it accelerate? Look at a certain population where the mutation rate hasn't been measured. Like, this is entirely possible, but the question is, why aren't they doing it? And I know I talked a lot there, but the reason why is because molecular clocks really are. We can trace back the history of humanity in our DNA. The clock ticks from generation to generation. So, yeah, go ahead. What are your thoughts on that, Snake?
Well, yeah, I mean, I wanted to read the stuff by Jensen before I comment too much on it. But again, we know that like there are multiple animals that have widely different mutation rates, and different parts of the genome mutate more often than other parts of the genome. Things like that. I believe like other animals were calculated to be much older than the 6,000 or 7,000 year old pedigree for humans using the same methods. And I wanted to get more into genetics a little later on. I still have a couple of
of course,
like morphology questions.
Hey, let me just make one last comment on that real quick, because for the most part, because I have a paper here that suggests that mammal-wise, most mutation rates are pretty well consistent. Now, I don't want to say constant because, yeah, they can fluctuate a little bit, but they're consistent. They're not 10 to 30 times slower in the past like the evolutionists say. Mitochondrial DNA, for example, ticks fast. These clocks are fast universally among all species. Now, here's the thing that's different. Generation times. That's why Jensen will look at the generation time, right? He can look at other factors as well, and that's where these they could look, for example, the Khoisan peoples, like some of the African people groups, their generation times seem to have been a lot different in the past. Therefore, that would reflect a slightly faster mutation rate, and that's why he's making a prediction to test whether or not that's true. And then, and before you get to that question, we can end it there on the mitochondrial DNA until you study that a little further. I'll send that to you. But are you then familiar with the Y chromosome now, with these papers that are suggesting that it mutates, it ticks faster than was ever expected? And now those Y chromosome differences, as fascinating as it is, only goes back just about 4,500 years, and we know exactly what happened 4,500 years. That was Noah. He would be our last Y chromosomal common ancestor, and those sequences found in the Y chromosome are nothing like the sequences found in the chimpanzee Y chromosome. The chimpanzee Y chromosome and human Y chromosome are like 70%, and there's some size difference as well. So it's less than 70% similar. So if our closest common ancestor is the chimpanzee, why is it nothing like the human Y chromosome? But the human Y chromosome, boom, goes back just 4,500 years. And Dr. Jensen said, okay, if this is true, there must be genetic signatures and markers in our Y chromosome, genetic stamps that can give us clues into the history of humanity. And boom, he's got like a 25-part series, highly technical. I can send that to you as well, where he's discovering all of these genetic signatures, migration patterns, things that should not be found if deep time evolution was true. Because remember, the last 4,500 years, according to deep time evolution and the Y chromosome and the mitochondrial DNA, that's just like the end, the tip of a needle of a long, long history. Those signatures shouldn't be there. It should just be a scrambled mess. So that's what I mean by genetics. Genetics is really, really, in my opinion, winning the war for creationists. Of course, you can comment on anything that I just said there, Snake. And then if you'd like to just move on to another question you had, I think regarding the fossil record, go ahead. Go ahead. Take your time.
Yeah, I mean, I'm more familiar with the Y chromosome stuff, and I've read a few things on pedigree-based mitochondrial mutation rates, and but I really wanted to read the Jensen stuff first
But what about
before I make a final comment on that. But as
the
for
Y
like
chromosome, though?
Y chromosome, yeah, it is more unstable, and
Right.
there's a bunch of research. Well, first of all, like if it wasn't unstable, then we would expect that to be the least common between the chimps and the humans. And then secondly, there is research on, I think, mole rats or some kind of rodent that is basically their Y chromosome is degraded almost completely, and what's happening is they're forming a new chromosome.
Is that
So
in the lab, that one?
that's just observing the genetics of the actual animals.
In those animals, like what was it that degraded to near extinction? A mouse, you said?
No, the Y chromosome in I think it was like a mole rat or something like that.
Okay, like this was in the wild, or was it like in a lab experiment?
It's just measuring wild ones.
Wild ones. Yeah,
Yeah.
I mean the point
So-
is with what they're finding is the Y chromosome does mutate a lot faster, because the thing is, evolutionists used to point, like there's one guy, Evo Grad, Rational Mind, he said regarding replacing Darwin, he says it's like Jensen just ignores the Y chromosome. He says nothing about it because the amount of DNA differences in the Y chromosome disproves young Earth creation, he says. But based on some of the newest studies and high quality Y chromosomal DNA sequences, it's now known that the Y chromosome changes a lot faster than we thought. Therefore, the results, and Dr. Jensen's put out recently a study, a peer-reviewed study in AIG, which I know you wouldn't agree with, but I'll send it to you anyway to see what your thoughts are. He demonstrated that the amount of DNA differences in the Y chromosome that exist in humans are only about 4,500 years worth of mutation accumulation. Now, here's the thing. If evolution was true, now we do know that Y chromosome is, I wouldn't say it's entirely the most unstable, because it is for the most part immune to recombination, right? Because it doesn't have a counterpart to exchange its genetic material with. Therefore, scrambling purposes, it's stable in a way where we should be able to look at the sequences themselves and look at markers, genetic markers, for example, which is what Jensen's doing, and seeing if he can detect historical signatures in the Y chromosome, which is what he's doing. So in that case, it's stable. It's immune to recombination. But according to the evolutionary model, if humans and chimps split nearly 6 million years ago, let's say, they would have had identical Y chromosomes at the split. Now, let's say the chimp line, okay, their Y chromosome was mutating fast. The human Y chromosome was mutating fast. Now we're here 6 million years later. So why was the chimp, the patterns and the population history of chimps so much different to humans that it still resulted in a Y chromosomal sequence difference of like less than 70%? Because that would have to be some really, really significantly different environmental histories, wouldn't you say?
Of course, humans would have been in a much different environment than chimps. They would have much different selection pressures, and if there's a small difference in just 4,500 years, then there's going to be a large difference in 6 million years.
Here's the thing, though. For that explanation to be, like, I've heard that, I think it was Rational Mind himself that said, Y chromosome differences could be explained by faster rates of gene conversion. But remember, you know what I'm going to say. What testable predictions can you make then? Right? What type of histories, what type of environmental conditions, when did the Y chromosome decay faster? When was it a little bit more stable, for example? I mean, anybody can just come up with these post hoc, ad hoc rescue devices and stories and stuff, right? And you would agree, you don't want to hear about what I believe. You want to hear about what's demonstrable, what's observable, what's testable. And that's why I'm saying, hey, listen, this is what the pedigree-based studies indicate. Here's the predictions that flow from it. evolutionists, they're kind of on the defense when it comes to molecular clocks. They're given reasons, they're giving stories and rescue devices, as far as I'm concerned. But without the testable predictions to flow from those explanations, they're just non-science. And I know you said you want to look at the papers and update yourself on it a little bit, so I don't want to beat a dead horse. Therefore, if you want at this point, and unless you have something else to say, we can move on to what other question you might have had. Go ahead, Snake.
Yeah, so I mean, I kind of wanted to circle back to genetics near the end, but kind of connect back a little bit. There's tons of testable predictions for other things too, so it's not like this is the only thing, and it's not exclusive to creationism either. But and I know you consider the fossil testable predictions to be less robust of a prediction, but one of the things that keeps me from understanding or believing the common designer type of thing is, like, to circle back to penguins. It seems like you're saying that penguins were not like micro evolved from a flying bird, that there was a penguin type population on the ark. And so if there's a common designer, then I don't understand why he's using, because he'll make fins, right? We have, what is a fin? But he'll use bird wing bones for the penguin fin, even though there's no reason to derive that from the other birds if they're created separately. And for whales, he'll use basically finger bones for their fin. And for sharks and fish, he'll use ray fins, kind of, for their fins. And they're all, it's an inconsistent design pattern for the same function and the same part. Meanwhile, the design of those parts fits more within an evolutionary pattern. And so that's one of the things that's really convincing for me personally.
Well, I guess sharks and fish are fish, whales are mammals. So you're going to expect similarities in places in morphology, anatomy, genetics, of course, ultimately. And with whales, fish, and sharks. And you're also going to expect dissimilarities as well. When it comes to the penguins, I mean, as long as there's a functional reason and a functional purpose for the trait you're looking at, or the morphology, for example, the bone structure. I mean, these things like homologous patterns, or just assuming God's design, assuming God's creation. I mean, we as engineers build weird looking designs. I mean, yeah, the patterns exist. The question is, are those patterns there for a purpose? Are they there for a function? Like, well, what's your biggest problem with the penguin again?
Well, it's like, why would you expect them to have similarities if God can just stick any old part on it, because he's created it separately? He has no reason to derive penguin fins from bird wings. And so the only reason you'd need homologous parts is under an evolutionary model.
Well, and that's the point. Like these shared structures, these shared patterns we see in the biological world, and we can even see shared patterns in the embryological stages as well. Just like you'd see shared patterns in the blueprint stages of an airplane, a boat, a sedan, a van, an SUV, but ultimately the final product has the most differences, of course. But the point is, like, homologous patterns of all types, that's expected on both models. And guess what? You know, if we're making that retrodiction, for example, and we're saying, okay, let's just look at the way we design things, okay? Because the Bible does say we're made in God's image. We want to know, how did God design this world? Did he use shared structures? Did he borrow structures, reuse them, for example? I mean, programmers reuse code all the time. It just makes sense, you know? And it's not like, like I said, we don't do it on purpose. We don't build in these homologous patterns. We don't build in these nested hierarchical patterns on purpose. It just makes sense from a design and engineering perspective. Therefore, that is an accurate retrodiction. And like I said before, Linnaeus, he's the father of taxonomy, and he based that classification, that taxonomic classification system, off a function. And evolution would explain those patterns as descent with modification. There's been changes, there's been additions, there's been modifying of existing structures. Like, I understand the theory, of course, but on a genetic level, that means our genetics, and we don't have to circle back to it now, but on a genetic level, that should reflect descent with modification. We should have evolutionary leftovers, genomic fossils. You look at the fossils in the fossil record, we should see fossils, genomic fossils in our genetics as well, that can connect us. But yeah, I don't know, like, I understand with what you're saying, but the thing is, that's like saying, hey, and that's why I pointed this out at the beginning, is you look at the sky and you say, hey, the sky is blue, therefore creation's true, or therefore evolution's true. No, those are the observations, those are the lines of evidence that are agnostic to the debate. And I understand that's convincing to you, but it also makes sense from a design perspective. Let's look to that which can differentiate between the evidence that is agnostic. And maybe you don't see it as being agnostic, but from an engineering perspective, it makes sense to reuse structures and parts, and we see it. We see it today. Go ahead, Snake.
Yeah, I don't see it as agnostic, because it's not reusing parts. If like, why would you put bird wing bones in the penguin when you could just put shark fins on it and make it super obvious that they're not derived from flying birds? Because you're not actually reusing parts. You're reusing the entire organism and making tiny changes. So it basically looks, even if God is creating them, it looks like in his laboratory he's taken a bird and just reduced its wings a little bit and made its feet smaller and better for swimming. Whereas if God could just put shark fins on it, and so
yeah, but you're assuming God's creation, man.
If he was reusing parts, all fins would have the same design, and things like why don't whales have gills? Like, there are swimming animals that have gills, so that they can survive in the water, and there are animals that have both lungs and gills. So it would be a superior design to have both. Like, a whale could use gills, and fish can use lungs, and some of the fish have lungs and gills, but it also happens that these lungs grow from the same embryological structures as swim bladders grow from, and so no fish that have lungs also have a swim bladder. So it's this weird thing where it's like, okay, well, it looks like the swim bladder, the thing that can hold air, is now absorbing oxygen slightly better than a swim bladder, and that's just a lung. Whereas a designer, if this stuff was just agnostic to the question, a designer, well, what we'd expect in a designer is he would give mammals gills if they lived in the water, and he could give lungfish swim bladders so that they could control their buoyancy, and he could give sharks lungs too. This would be no problem for him. But instead, what we see is all of these traits are nested within evolutionary hierarchies. Like,
well, and there's where my problem is yeah, just like I can point to the fact that modes of transportations, you can even do it in computer code, for example, but you have options, sub options, for example. But I'm not a programmer, so I like to use modes of transportation. You can look at sedans, which have a vast amount of similarities with other sedans. Bring in a semi-tractor trailer, for example. That is a lot different than a sedan or a car, but those two modes of transportation, you can bring in SUVs, vans, boats, airplanes. These are all different enough, but yet similar enough to each other that it sets them apart from other modes of transportation. Like, you see these modes, you see these groups within groups patterns within things that humans design. Now, here's the thing, though. Yeah, we see these patterns in the design world. We see these patterns in the biological world. But there are many instances where the patterns or the features don't actually follow a reasonable evolutionary history, for example. That's why anytime there is an inconsistency or lack of uniqueness in the tree, in the phylogenetic tree, convergent evolution is invoked. So it doesn't all just fit perfectly. And yeah, you can assume the design, but let's say God created these basic kinds. He created them with pre-existing functional DNA differences, okay? Created populations, created them in bulk, for example. If you came at the creation event as a scientist and started looking at morphology, anatomy, genetics, for example, you're going to see nested hierarchical patterns on every single level. That's going to reflect universal common descent that you're looking at now, when the fact is, in that moment, an hour after creation, that is, those patterns are there for functional purposes. Now, for example,
you see breaks in that pattern, like a gill on a
how
whale.
would you see? No, because the way God created the initial created kinds, now we would see speciation events. Since then, we would see changes in allele frequencies in these populations over time, for example. But you're never going to see anything that would be inconsistent with really even with what evolution would expect today in changes. I mean, yeah, you're not going to see like some Pokemon like Bulbasaur with a plant on his head or something like that. Like these
Well, why
initial
not?
created
Why couldn't God do that?
Because if God didn't create a form of life that reflected that type of morphology and anatomy, well, then that's never going to suddenly occur in the history and the population histories of these animals. Like I said, these biblical created kinds are going to speciate and change in the same way and in the same mechanisms that even evolution would propose. That's why these patterns in general, now you see these patterns in the fossil record, you would see them hypothetically at creation, because those pre-existing DNA differences that we would hypothesize were front-loaded in Adam and Eve, and that applies universally across species, those necessitate nested hierarchical patterns. But with you, and as I was saying, if you look at, let's say you're looking at the outward similarity, okay? Look at marsupial moles and look at placental moles. They're not classified together. What they do is they take the marsupial mole and they group them with creatures like kangaroos. And then the placental moles, as far as I know, from my understanding, they're grouped with animals such as llamas. Therefore, despite their outward resemblance, okay, these types of creatures belong to very different taxonomic categories. And that's what I'm trying to say is when convergent evolution or loss of traits evolution or incomplete lineage sorting is invoked, they're admitting, okay, this specific part or this specific feature or trait does not follow the expected evolutionary taxonomy. I mean, those examples exist. I mean, I can go on for hours and hours about these types of examples, but the rule is those patterns exist, but you make it out to be like it's perfect. And the hierarchies you'd see in modes of transportation, they're not perfect, right? You can point to inconsistencies there as well, just like the hierarchical patterns in the biological world are not perfect. But a line of evidence that can differentiate between it is exactly what evolutionary biologists look to today. Like, for example, Grar in his paper where he fights so hard the function of the genome. He's the one that said, if ENCODE is right, evolution is wrong. And therefore, he has this extensive paper showing why the genome, both protein coding and non-coding DNA, can't be any more than 20, 25% functional, because the genome has to reflect what you're saying. But if the genome reflects hierarchies by design, well, there's another line of differentiating evidence that can go hand in hand with the molecular clock. So I understand you probably look at this line of evidence with that basic assumption and say, okay, the patterns, the homology, the mosaics, they reflect descent. Here's the thing, it's neutral because we expect the same thing. You got to go with that which can differentiate between the model and DNA function. Two totally different expectations. That's why the evolutionary community fights so hard, a genome of function. But I know I said a lot there, so just touch on anything you feel necessary that I said, Snake. Go ahead.
Yeah, I mean, the marsupial and placental moles are still very highly closely related. They're derived from a small, furry four-legged mammal, and like, yeah, there are like two-legged marsupials like the kangaroo, but it's still got all of the same parts. They're just like in slightly different proportions. It's got slightly larger back legs. So that's not really a breakage of.
I'm not saying there's a, yeah. When it comes to nested hierarchical patterns, you can look at the most you can look at morphology, anatomy, genetics. You can look at the highly conserved mitochondrial DNA proteins like the cytochrome C. You can look at amino acid trees, neutral variation in third position codon redundancy like Herman Mayes does. Hierarchical patterns are the rule. That's exactly what we see. I'm just saying there are some inconsistencies. The inconsistencies evolutionists sometimes confuse with like a complete breakdown in the phylogeny. No. The hierarchies do exist. That's the rule, but the question is, do both models expect such a thing? And if they do, what can differentiate between those expectations? I'm just pointing out inconsistencies, but of course you can still fit them somewhere because all forms of life share anatomy, morphology, genetics. I mean, you can take something like a skateboard and a sedan, so dissimilar, but you're still going to find hierarchies based on materials used, based on the materials in the wheels or the tires. There's always going to be hierarchies. That's just what's expected based on descent with modification and based on created heterozygosity hypothesis. The thing is, though, we've already made a prediction as to how we can determine which one is true, and we've made very specific predictions on like the cytochrome C protein and just DNA function in general. So go ahead, Snake.
But if God can create anything, we should expect to see a lot more breaks. Like there's actually no reason to have that hierarchical pattern. You might see it here and there, like, okay, like there's two things with like a bird head or something like that, or the platypus has the duck bill. You would expect to see that occasionally, but you should expect to see way more diversity, like a whale with gills or a Bulbasaur. And that kind of leads me into like real quick, and then I want to get back into genetics. Like,
yeah, take your time.
So basically, like where I started with all of this is like I don't understand why something like whale evolution would not be possible even under creationist model. So you've probably heard the spiel before, but to go over it really quickly again for the audience, if you have like a dog type animal, maybe that has hoofs, and it starts swimming in the water, and it starts losing its hair, its nostrils get a little higher on the skull, its padded feet get a little more spread out, and it loses its hind limbs, and it gets a flatter tail. That doesn't seem like it's breaking any of the creationist genetics or evolutionary rules, because there aren't actually any new traits forming, and yet just by the changing of the traits that are already there, you get an entirely different morphology. So is that impossible to you, or is that just something that just happened to not happen?
So
or is there like not enough time for that to happen?
Right. Okay. So my problem would be, and I believe it was Dr. Jonathan Wells who went over, countless changes that would be necessary. You're looking at the forelimbs would have to be changed into flippers, the modification of the eye for underwater vision.
Well, the AIG already says that happened with otters.
Well,
weasels.
Who says that? Which example? The forelimbs?
Well, AIG has said that otters are a type of weasel, and they're in the same kind, and yet we see from otters that some of them have started to lose hair on their tails. Their tails get flatter. Their limbs are more paddle-like, especially their hind limbs. Their eyes are evolved for swimming. I think some have what is it? The reniculate kidneys. Bears have that too.
Right, but I'm saying
so they're all just changes to existing anatomy that It doesn't
Right, of
require any
course.
actual new
It's
traits.
just modification of existing traits, features, structures. I understand all of that, but I'm saying this list that you can see and things that the evolutionary community would even agree with. I mean, just in general, the origin of tail flukes and the musculature associated with it. I mean, I can go on and on. I mean, the ability to nurse young underwater. I'd have to see the example from AIG that you're talking about and see what they conclude is the same kind. But I believe it's only like one. I mean, it's only several million years for all of this to occur within a land dwelling cetacean and the first truly aquatic whales. And even when you look at, because I always point out the fact that the fossil record is so interpretable that it can almost fit any story that you want it to. The Pakicetus, when they first discovered Pakicetus, they illustrated it as some fully aquatic mammal. I mean, you can see some pictures, you can see some writings on it, but there's been a huge change since then. And now they've been based on further findings, other fossils, for example, they portray it as just an ordinary land mammal, which is exactly what it was. But the same thing goes for Ambulocetus, Rhodocetus, Basilosaurus. I mean, you can look at all of these, and even I mean, do you disagree that those bones that they say are the remnants of legs, for example, the walking whales? Are you denying the fact that they use those for reproductive purposes? I mean, their reproductive organs are very large, and then they would need, assistance and muscles, for example, to work those. So even that
Well,
example I find to be flawed. Go ahead, Snake.
well, some whales do, but then some whales don't have them at all, and so they don't need them apparently. So God could have just designed them either way.
Well, I don't know.
So it
The
doesn't
paper
actually
that
matter. But all whales in vitro develop hind legs and then lose them, and same with whiskers
yeah,
and
I
then
talked
lose
about
them. There's absolutely no reason that would happen under a design model.
Well, I talked about that earlier. The fact that you do see similarities in the in those stages of embryo of embryonic development, right? Everything seems you can put a reptile next to a mammal, next to a bird, next to a human. You can see similar um, stages in development, but once you see that fully formed organism, that's where you see the major differences. Just like, and once again, this goes to the neutral aspects of the debate. Just when we look at the blueprints, you can line up a sedan with a boat, with an airplane, with a van and SUV. You're going to see in the blueprints, you're going to see that everything is nearly similar in the first stages or the second stages or the third stages, but it's not until the end products that you see the major differences. And that's exactly what we see in the embryo in these different stages of development. So once again, if we're made in the image of God, let's look at the way we design things, not only the blueprints, but the finished structures. For example, we can look at computer programming, we can look at modes of transportation. These are the agnostic
But
lines of evidence. So sorry, go ahead. Oh, no, I just wanted to point out one thing. So the paper that I have, it's not a creationist paper. It says new study turns a long accepted evolutionary assumption on its head, finding that whale pelvic bones play a key role in mating. And there's an article associated with it that says whale reproduction, it's all in the hips. So I mean, if it's assumed that some of the smaller whales don't have it, well, no, it's probably there. But I mean, if you're looking at some of the larger whales, like a blue whale, it's going to be more pronounced, of course. But you see those same structures in the fossil record in the so-called transitions. And I would just assume that they're also used for reproduction. I mean, from a creationist perspective, it makes sense. So go ahead there, Snake.
But if embryonic development was designed, why are they adding parts that fit the evolutionary pattern? They're growing legs and then losing them. Like, we don't see that with any type of design. Like, we don't put parts on cars in the factory and then later just take them off because they
Well,
had
no,
no
no,
function. That doesn't happen.
no. The blueprints reflect the same thing. You can make the exact same case in the blueprints of these various modes of transportation that I just laid out. I'm not going to reiterate it for sake of time, but you can make the exact same cases you're making right now, Snake. I could make, you could make, anybody could make, based on the designed world. We see the so-called transitional forms. We see the nested hierarchical patterns. We see the similarities in the blueprints, in the designs themselves. And you can look at computer code and do the exact same thing. So it's just expected on both models. You know, I don't know what to tell you, but the differentiating lines of evidence, like your molecular clocks, the predictions, what predictions, especially because you're right, predictions in the fossil record, since it's indirect evidence for ancestry anyways, and oftentimes, I mean, how would you define a species just looking at some bones, some fossils?
Anything that seems to have some kind of breeding barrier that
From how are you going to know if a bunch of like if you found these 450 breeds of dogs we have today, or the over 700 breeds of horses, you know, there's a lot of variation just in those breeds. If those were, look at the bovid, look at that family, massive. Deers, for example, huge variation in these species. So if those all went extinct, they were buried in some local flood, say, and they're dug up thousands of years later, I mean, you're not going to know what can breed. How are you going to make those assumptions as to what can breed with each other if there's no genetics? Go ahead.
Well, but that's the thing. But what I was talking about with the embryonic thing is there's no design in which we just start building like 6 wheels on a Volkswagen and then take those off later. But
well, I mean, you were the one saying that the transition from a small dog-like mammal to a whale resulted in the loss of structures. Isn't that what you said? I mean, you even admitted that there wouldn't really, and that's exactly what we see.
In development, like when a whale is is growing in the womb, it adds its hind limbs, like the actual flesh, and then those disappear. So that would be like adding on 6 wheels to a car and then taking off the 6 wheels.
I'd actually have to.
There is more variation. There is a lot of variation within kind, and that's kind of the thing. Is like we do oftentimes go, okay, we thought that these fossils were separate species, but maybe now they're the same thing, or we thought that they were the same thing, but we found something that's a lot different. So they probably had separate populations, but the overall similarity is like, okay, so these were probably derived from a similar organism that had the shared traits that they have in common. And and when we look at any of the fossils, I don't see any reason why that's out of the picture, because we know that bones move, that bones can duplicate and move and
but how can
change size and change orientation.
How can you determine? So, like, I don't really think your definition for species in fossils was a scientific or even reasonable definition. How are you going to determine what similarities are due to common ancestry, or which ones are just due to, like, let's say, convergent evolution, for example? I mean, look, how can you define if fossils are so open to and what you're saying when it comes to the similar stages of manufacturing, that all has to do with efficiency, and it also has to do with function. So the shared elements that you see, let's say we were looking at even not just different modes of transportations like SUVs, vans, airplanes, boats. Let's look at different companies, Honda, Chevy, and Mercedes. They're all going to have shared elements located in the blueprints. But I think with what you're saying with the whales, for example, and other different species, I think the interpretation is that these structures are actually appearing and losing for no purpose and no function, kind of like the so-called gill slits that the evolutionists will say with Haeckel's drawings, right? They'll just say that those really are some type of remnant of when our fish ancestors, when in fact those go on to form an important purposeful structure You know what I mean? So I think that that's just comes down to interpretation. Like I said, you can take those shared elements and blueprints of different manufacturing companies, come to the exact same interpretations and conclusions. But I'm telling you, it's not evidence that's going to differentiate between the model. I mean, you got to look to genetics and you got to look to predictions. What predictions can flow from these? Because I don't agree that these so-called transitional forms that were predicted, you see that they suffer from the grandfather paradox. Loss of traits evolution is always invoked. They're not like predicted in this layer in this spot, and that's exactly where they're found. Not even, I'm not sure who told you that, but get your money back. Because I mean, they're always changing around the story. It assumes deep time evolutionary history, molecular clocks proves that false. It assumes away a global flood, for example, or it assumes away burial by ecosystems. And I mean, that's getting into a whole nother debate. But if you want to make another couple points on like fossils, for example, and whales and stuff, you can. And then if you want, we can go back into, well, actually, let me see how much time we have so we can, because we're going to be going on an hour and a half soon. I know we wanted to get into the next stream. So, you know what, take this away uninterrupted. Any points you feel you want to address, anything you feel you want to say that you haven't been able to say, go ahead, Snake. Take your time.
Well, again, like with the parts, the gill parts probably do become important in the human anatomy, but the whiskers and the legs and the whales don't. They're entirely lost in the embryonic development. And so basically, I guess what I wanted to get to, there was a lot you said there, so I'm trying to gather my thoughts.
No, I'm not going to take your time.
So, I mean, when you said
I'm going to listen. Really take your time, though, because I got to run upstairs for 1 second. I'm going to listen from my phone. Go.
Okay, so you'll hear it.
I'll hear it.
Okay. So when you were talking about loss of traits and like the grandfather effect, I mean, that's all accounted for. The grandfather effect definitely is accounted for in evolution, because if we have a population of fish that can go on land, you're going to have some fish that are going on land and they remain fish, and then you're going to have some that split off and get better at walking on land. And then if that original population still exists, or multiple populations of fish that can go on land and back into the water exists, then, you know, later on, that's also going to happen as well, even though an early population has already maybe stayed on land. And like with the Pakicetus, we might have thought that it was mainly a swimming animal, but then thought it was mainly a land animal. But these details don't matter that much, because we know that it at least swam sometimes. And this is kind of like the otters and the weasels, whereas their AIG claims that they are the same biblical kind, and yet there is clearly a lineage of weasels that has gone off and adapted to swimming primarily in the water, and they look a lot like seals and early whales would have. And the point about the predictions of the transitional forms is that not just that we can line up the bones, but that they're in the precise position that they should be if they were in a transitional lineage. And I guess finally, the point about loss of traits always being invoked, that's invoked a lot when I talk to creationists, because that's all they'll accept. And so that's pretty much, we'll try and work within that and show how a kind can change even within loss of traits. And AIG will even admit this, that snakes could have come from lizards. And so that is an admission that macroevolution is at least possible. But and I know we're running short on time, but I wanted to touch on bird evolution really quickly, how that doesn't rely on a loss of traits, that we actually have a lot of evidence that feathers are scales, and dinosaurs had feathers already. And so basically a bird is just a dinosaur that had a calcified face, like for a beak, or not calcified, but keratinized, and that they just got more feathers. And there's a lot of evidence that feathers are scales because they're made out of the same cells, the same protein. It's just that the cells die in a certain pattern so that there is a fan pattern. And we've done this. We can just add in a simple retinoic acid to chicken feet, and instead of scales, they'll grow feathers on the feet. And a very similar branching structure can be grown from alligator scales. So the process to create new traits from old ones is already there. And then there probably was another point in there, but I think I've covered most of the stuff that I wanted to, and then we can get into a couple of the genetics stuff real quick.
Yeah, you've done good. You've done good. This has been a good discussion.
Okay.
As always, it flies by. I think we've touched on a lot of good things.
I know. Where's the time go?
Where's the time go? I tell you. So I would just say that I'm not sure if we're just going to go in circles when it comes to the shared elements and blueprints, homologous patterns, nested hierarchical patterns in anatomy, morphology, and physiology, as well as I find that you're just assuming God's creation as if he would create something like a Bulbasaur. If this wasn't part of the original creation, if this wasn't a reflection of the original hierarchy, it's not something that we would expect. But yeah, those lines of evidence, including the transitional forms, I would just say are agnostic to the debate, because from a design model and a design perspective, it's exactly what we would expect. And I always point to the father of taxonomy, like Linnaeus: kingdom, phylum, class, order, family, genus, species, based on function. He was a creationist. We're the ones making the predictions on function anyways, that those patterns are there for functional purposes, of course. When it comes to the snakes, from my understanding, the creation organizations, AIG, ICR, Creation Ministries, they would say that in the pre-flood world, there could have been some snakes with legs. I mean, we know the serpent in the Garden of Eden was cursed. And there's evidence in our genetics that there's some broken genes regarding legs in snakes. And therefore, that curse could have actually been a genetic related curse, an epigenetic related curse. I know there's a genus of snakes, I think Najash, that debated a herpetologist, Adam Heat, many times. And we discussed snakes for 20 minutes, to be honest with you. So I'd probably just tell the audience to go watch that again, so we don't hash that out for 20 minutes. But I find snakes to be fairly interesting, and I also find the theory that the curse was based on either epigenetics or genetics, where those legs were lost at some point pre-flood, and now today we see snakes with no legs, of course. When it comes to the birds, dino to bird, I would just go back to the fact that what I was saying in the beginning when it comes to species. So we're also making predictions not only on mutation rates, DNA function, but also speciation rates. The amount of bird species today, 10 to 12,000. Okay, that makes sense coming from a handful of bird kinds from the ark. We can actually make testable predictions from those numbers. If, as you're saying, modern birds evolved from theropod-like dinosaurs millions of years ago, I assume roughly 65 million years ago, why are there not 500,000 species of birds? Why are there only between 10 to 12,000? I do want you to answer that, but I do want to touch on one point that oftentimes the evolutionists will just point out once again the similarities in morphology, anatomy, and physiology with birds and theropod-like dinosaurs, but there are a ton of distinctive features that can distinguish birds from dinosaurs. I mean, birds are homeothermic with a high metabolic rate and a high body core temperature. They've got a unique respiratory system. It's very efficient. I believe they refer to it as a high efficiency flow through respiratory system with no diaphragm muscle, numerous large air sacs. I mean, I can go over a ton of differentiating features and traits from birds and theropod-like dinosaurs. But my biggest problem and concern is the number of species that we see today. So how would you? Yeah, go ahead. How would you answer that from a deep time evolution starting point?
Well, there are extant and extinct reptile species that have unidirectional airflow and hollow bones and thermoregulation. Even species that exist today, like turtles, there are like turtles that are like medium warm-blooded, and there are I think crocodilians and lizards that have unidirectional airflow lungs like birds would have, and there are plenty of dinosaurs that have the same type of structures that we associate with those types of anatomies, and they also have hollow bones and things like that.
Well, like lungs and diaphragms being soft tissues, they're I'll say infrequently fossilized. So I'm guessing a lot of the things you're saying would be more so inference based on the starting point that birds did evolve from theropod-like dinosaurs. Because
it's based on the similarity to the reptiles that are still alive today that have that. So either they evolved that convergently with birds, or back then the reptiles had those traits in the population already.
But doesn't the fossil evidence demonstrate that theropod dinosaurs, which, as you're saying, is what is believed to be the ancestors of modern birds today, didn't they have lungs that were more so like living crocodiles and alligators, crocodilians?
Well, there are crocodilians with unidirectional airflow.
Right. So if the theropod dinosaurs that are proposed to have evolved into modern birds today, the fossil evidence suggests that they had lungs that were more similar to crocodiles and alligators and not modern birds that they're supposed to have evolved into.
But they have both, so both of those traits would be in the populations, would be possible in the populations.
So my point is you can look to the similarities all you want, but there always are dissimilarities. There always exists distinguishing features, traits in not only genetics, but anatomy and morphology, and the patterns exist, for example. But like I said, my biggest concern would be for one, obviously the novelty that would have to take place to take your dino to bird, especially when what we observe in the form of mutations is reduction as well as compromise. For example, a lot of these mutations, the changes seen are functionally compromised. So I don't see how that's going to take things forward. But yeah, how do you explain the number of species that we see today in birds that correlate nicely with a young Earth creation model of speciation post-flood versus the so-called deep time, millions of year history of dino to bird? Go ahead.
Well, there's actually a creationist technique that I don't think it's used much anymore because we have a lot more fossils, but it's called taxon correlation. And basically they make a three-dimensional graph and cluster morphological similarity. And so they've done this, a secular study took this creationist method and did this updated with more modern fossils that we have, and added in the theropod dinosaurs and the avians and proavians. And so basically the method goes, if there's more distance between a point or a cluster, then there is distance across the cluster, like within the cluster. That means that it's a separate biblical kind. But what they found is that there is a smaller distance between some of the theropod dinosaurs and the birds than there is between theropod dinosaurs and other theropod dinosaurs.
Yeah, clustering. Yeah. For example, Todd Wood does that. you have to look at anatomy and the outward appearance and morphology for the most part. So people like Dr. Jensen, Dr. Carter, Dr. Sanford, myself, Ramat, for example, we would disagree with that approach. Ancestry should be determined through genetics, because that's what's inherited sperm and egg. It's the only direct way of determining ancestry. So I disagree for the most part with the clustering technique as to how we can determine relationship and kinds, because of those patterns. Those nested hierarchical patterns make it difficult to, like, for example, we would even say humans, the great apes, pigs, and say ducks are all different kinds, but because they share hierarchies in anatomy, morphology, and genetics, even between, say, a duck and a human, you're always going to be able to draw a line somewhere that seems to indicate relationship. Just like you could, you can form relationships, as silly as it sounds, with a skateboard, a boat, and a sedan, because patterns exist. So yeah, the clustering technique, looking at morphology and anatomy, that can oftentimes fail us, as I've stated. So molecular clocks really do show, at least, especially with humans, based on mitochondrial DNA, Y chromosome, that humans are their separate kind, humankind, and DNA function, especially when it comes to all the other forms of life that, say, we're looking to now. So yeah, I do agree. I do know of that technique, but did you answer the species, like the number of species question, Snake?
That there's a small amount of birds.
Yeah, well, and it's not even really just because we're talking about birds, but you can look at bats, you can look at cats, you can look at lizards, for example, snakes. I mean, there's only a handful of species of all these different.
I'm pretty sure bats have the most mammal species of any mammal.
Yeah, and it's still not even that much. I think it's like maybe 2,500, which is easily explainable since the flood 4,500 years ago. So when it comes to birds, I mean, according to your model, like with what you're saying here, modern birds evolved from theropod-like dinosaurs millions and millions of years ago. Why are there not 500,000 bird species? Why so few? Why does the amount of species seem to correlate better with young Earth creation timeframes? When it comes to with the bats, how many bat species are there? I think there's 20, 25, 26, 2,600 maybe. There's really only like 1.6 million species on Earth. If you actually want to just consider mammals, reptiles, amphibians, and birds, there's between probably like 30 and 40,000. Insects constitute most of them, like 1 million species. But yeah, I mean, the number of species correlates better with the young Earth creation model. So yeah, from your the bird. Let's just focus on birds.
Dispute that.
Bird species.
Well, I would dispute that. I mean, I didn't look into the number of species in each bird kind as much, but I know with the reptile kind, the amount of species per kind, according to AIG, would range from 1 to like upwards of like 600. So I don't think that that's consistent at all.
How many reptile species exist?
I'm not sure. Thousands of them.
Yeah, I think there's less than 5,000, maybe 4,600. I mean, I'm going at the top. I should pull up the data, but once again, that correlates way better with a young Earth creation timescale of 4,500 years since the flood. Handful of reptile kinds come off the ark based on those front-loaded pre-existing DNA differences, gene conversion, recombination, genetic drift, isolation, inbreeding, for example.
But what are you expecting? Are you expecting all kinds to have similar amounts of species?
Yeah, well, I mean, the predictions that are being made, I've got a highly technical paper here, 30, 40 pages from Dr. Jensen, where he goes into all these different species and shows that the species are originating based on linear changes, consistent speciation rates. So, I mean, don't you find it kind of odd that there's only between 10 and 12,000 bird species today, if deep time evolution is true? I mean, 65 million years ago, theropod dinosaurs started evolving, or they did evolve into your birds. So how many extinction events has there been to reduce the number of species today to only like 12,000 roughly?
Well, it doesn't take specific large speciation events, but I'm not sure what you would be expecting in the first place. Like, could you email me that paper too?
Yes, yeah. We'll email that. Okay, well, instead of going in circles, did you want to? We've touched on a lot of good points here. It's been fun. We're now at an hour and 35 minutes, but did you want to go into? Is there any other points you wanted to make in genetics, or is there any last things you wanted to say, or something you wanted to touch on? I mean, I know I've said many times that the function of our DNA and the DNA element and the markers, especially. We would predict that those are highly functional if the hierarchical patterns are there for design purposes. So I've talked to many evolutionists that'll admit, yeah, the majority of the genome should be based on evolutionary left. Have you read the Grar paper where he tries to make the case that the genome is no more than 20-25% functional, or else we would have gone extinct?
Yeah.
Yeah. Oh, yeah. We've talked about that before.
Yeah, and so I have a couple questions. Maybe I can state a few of my grievances, like maybe in a closing or something. But I'll try and get to a couple of them while I have you. So for kind of in reference to what you just said, if every genome was needed to be mostly functional, that doesn't quite make sense to me for a couple of reasons. I mean, one, that paper makes sense, but it's talking about functionality in a very specific sense, in that it's a coding gene. There are obviously functions like
it does refer to. I feel like that's a mistake you often make. No, they're considering like, so for example, in the non-coding regions, what some have called junk. Yeah, they've always expected some regulatory functions.
Yeah.
Some functions in the retrotransposons, the pseudogenes, some though. They've never predicted any more, just like with Grar said, any more than 20-25%. So you need to understand, and I'm 100% certain on this, especially after talking with like Frello and Mays, Conspiracy Cats. This includes non-coding and protein coding, because only, is it 2% of our genome codes for proteins. The rest of it, which is the non-protein coding DNA, that's doing other jobs. And I would say that the non-protein coding RNAs are regulating virtually all aspects of the gene expression pathway. But the evolutionist like Grar, he would disagree with what I'm saying. So there really are two separate expectations and predictions here. So no, he's considering the non-coding regions too, Snake. I mean, would you disagree with that?
That's the upper limit to the functional fraction paper, functional fraction of the human genome.
Yeah, because remember, he's the one that said if ENCODE is right, evolution is wrong. But ENCODE is what revealed through biochemical activity, right? They defined function as specific biochemical activity. They're the ones that said over 80% of our genome is active and transcribed into RNA. Well, RNA, DNA, RNA, protein, of course, not every time is the end product a protein. Oftentimes it just goes DNA transcribed into RNA, and then those RNAs have a variety of jobs. So that was the surprise there. So yeah, he's considering the non-protein coding regions. That's the majority of the genome for the most part.
Well, they're just considering basically necessary functions. They define it as a capacity that has been shaped by and is maintained by natural selection.
Right. Like, well, I mean, conserved regions would be maintained by.
So there would be regions that would have a biochemical function, but are not selected for, basically.
Right.
So, yeah. But basically, I mean, the larger point I was going to bring up is,
take your time.
Like, so there's this thing called the onion test where we look at, some onions have twice the size of a genome as other onions. And so we know for a fact that at least about half of the genome of the larger genome onions, that's not all functional. And things like amoebas some amoebas have the largest genomes out of any organisms, period. So either it took God more it takes more information for God to make an amoeba, a single-celled organism, than it takes for God to make a human. Either that, or a lot of the DNA in the amoeba is basically not used.
Well, here's my response to that. So, and good point. The onion problem is definitely a good point to make. Plant genomes, though, they are larger for adaptive purposes. So think about it, Snake. Plants can't move. Okay, they can't put on a jacket if it's cold out. Okay, so if you're looking at temperature fluctuations, pH changes, for example, acid rain, for example, you name it. Okay, so plants are they are constantly facing negative environmental changes. Okay, and it can't do anything, so it has to adapt quickly, or it dies. So therefore, even like an animal, if you look at certain animals that can adapt, they can move underground. For example, humans, we can go in our house, put a coat on. So they need bigger genomes for redundant purposes, backup redundancy. And here's my question to you. I find redundancy to be fascinating evidence for design, because natural selection, which you were talking about, how it preserves these conserved regions because they're important. Natural selection has to do so. Natural selection works in the short term. It doesn't work in the long term. Therefore, how do evolutionary processes like natural selection build those mechanisms, like DNA repair mechanisms, redundant based mechanisms, for example, that work in the long term? How do you account for that?
How do we account for DNA repair?
So, for example, redundancy. So my answer to the
I
larger
mean,
plant
that's
genomes is because they need more redundancy. But natural selection works in the short term, not the long term. Redundancy is
but that
beneficial for the long term. How does natural selection evolve that redundant mechanisms and DNA repair mechanisms? Go ahead.
that's one of the functions of so-called junk DNA. Is it's not junk in the sense that it's garbage. It's
How does it
just,
evolve that?
how did it evolve it?
How does natural selection evolve that which is necessary and beneficial for the long term? Natural selection sees the short term. How is natural
Well,
selection building a spare tire, essentially?
yeah. So it's in the theory, it would be stuff that's like kept in the background in order for future adaptations.
So it knew.
Stuff that can be
It
assembled. So how does
knew.
that get selected for? Is, I mean, we've observed things like fruit flies and things expanding their genome, and it's basically loss of features happens a lot, and so you don't need every gene that's in the genome. So you'll lose some of the genes, but it's not that it gets cut out because it doesn't intelligently know, okay, we don't need to use that gene anymore. We'll just get rid of it completely. It's just that it kind of gets shuffled to the side. It doesn't get used anymore, but the instructions are still there, and it might be turned off and it might be deregulated. But most of the stuff is still there. And so natural selection doesn't know to just take out all the stuff that's not being used at the time. So it's just the way it works.
Right, but it's like, so for example, when we build cars with a jack or a spare tire, which to an evolutionary biologist, they would assume that that spare tire is the result of an evolutionary leftover. Or in computer code, we redundancy is considered good design. Backup code is beneficial. And then we see the exact same thing in the DNA code. We see redundant mechanisms. We see a need for more redundancy in plants. So that solves the onion problem. And the evolutionist now has to concoct a story as to how natural selection, which, as I said, sees the short term, not the long term, could evolve such elements and mechanisms that are clearly built for backup and beneficial purposes. But this type of multi-dimensional codes and these multiple layers of mechanisms and elements and whatever complexity in the genome, it's exactly what we would expect. Like, for example, one thing that fascinates me is alternative splicing. The fact that a single protein region, okay, of DNA can actually give rise to hundreds and sometimes thousands of different proteins. I mean, that's another thing I'm baffled as to how evolutionary mechanisms like natural selection can evolve such a thing. I mean, how would you explain alternative splicing, for example?
Well, if the transcription factor just binds slightly down the line, then it's going to be transcribing different parts. And I mean, if you translate something into protein, it's probably going to do something. There are very few inert proteins. So if it has a beneficial function, it's going to be conserved. But I mean, I've always said that evolution isn't incompatible with intelligent design. It's just not compatible with like young Earth creationism. So God certainly, if He created life, certainly would have put in the capacity for junk DNA if it was in His design that things should be able to evolve. If it wasn't in His design that things should be able to evolve, I would think that He would give us less of a capacity to adapt and would restrict the genome to like strictly 100% just what is necessary. And then whatever happens to that, well, that's just part of the fall. That's degradation and genetic entropy.
Well, when you get into biblical theology, it would make sense through God's foreknowledge. He would know we would have fallen, and therefore he would have built our genetics with redundant and DNA repair related mechanisms. But like you said, yeah, less than 2% of the DNA codes for proteins, and those proteins are functional and even multifunctional. For example, the protein moonlighting phenomenon, which is what we're looking to in making predictions on these hierarchical patterns in like cytochrome C, these ATP synthase proteins, for example. But guess what? What's the rest doing? This is the big question, and this is where evolutionists have to account for all that as mostly evolutionary leftovers. Yeah, some of it's involved in regulation, but the majority of it is just there. It's just neutral junk for the most part. Maybe a hidden reservoir for evolutionary advancement in the future, like you were explaining earlier. But it actually turns out that the rest of it, the other 98% of the genome, is a huge control panel. You've got millions and millions of switches that turn protein producing genes on or off. I always explain it like if you go to a theater and you're watching a live show, for example, you got all your actors on the stage. There's a lot going on behind the scenes, and it looks like that this 98% of the genome, that's exactly what this is. They're there for a purpose, there for a function, and this is a testable prediction that we've made based on the created heterozygosity hypothesis. So if the vast majority of these DNA elements, redundant mechanisms, the genetics really is mostly functional, that turns those patterns that we've been talking about for an hour and a half into design patterns. Evolution, for evolution to be true, descent with modification, the majority really does have to be based on genomic fossils, evolutionary leftovers. Well, you mentioned conserved regions. They were shocked that there were so many conserved regions. For example, even between like the mouse genome and the human genome, you have these conserved regions that, you're right, natural selection preserves these because they're functional. If deep time evolution was true, and we've separated from obviously common ancestors with something as far on the tree as like mice, why are there so many conserved regions? Why isn't it more of a scrambled mess?
Well, I mean, there are some conserved proteins, but oftentimes you'll see that, again, they follow the evolutionary pattern, whereas like cytochrome C, it creates the same protein, but the sequence is much different in worms, whereas the sequence is exactly the same in humans and chimps. It makes the same protein, but we can see that it's been accumulating and following a lineage pattern rather than just an intelligent pattern. Whereas if humans and chimps have been evolving the same amount of time, we would expect to see the same amount of differences between each kind. I did have, I don't mean to like rush you or anything, but I did have a couple of questions.
Of course, yeah, of course. Let me just comment on the cytochrome C example, because yeah, those are considered housekeeping genes because they're necessary. They're absolutely required for the maintenance of cellular functions. For if they're not there, the organism will die. The existence of the cell will not be there. So, like you said, and yeah, that is getting into some of the stronger evidence for universal common descent. Those highly conserved mitochondrial DNA proteins, like even the CO1 gene, where you can look at DNA barcoding, these all fall within nested hierarchical patterns. But the thing is, the difference is, like, for example, the cytochrome C protein, those ATP synthase proteins in humans and chimps are nearly identical, like you said. But if you go down to like the old world monkeys, go down to dogs, fish, for example, mice, those highly conserved mitochondrial DNA proteins, they're different enough where they reflect descent with modification. But the question is, are those differences in those sequences a reflection of descent with modification or reflection of design and function? And that's why I was explaining the protein moonlighting, these proteins that have multiple functions. It's like a multifunctional tool. You got 10 different functions on these tools. I mean, this is science fiction. So we're taking that prediction. Jensen took that prediction on nuclear DNA proteins that have multiple functions, and he's saying that those differences in, say, your cytochrome C protein also reflect function that is specific, not only multifunctional, but functional specific to the kind of organism, right? Like the secondary function for something like an ATP synthase protein will be different in say metabolic optimization or cell optimization in a human and a fish, and that'll reflect those patterns. But guess what? Right now they haven't done the experiments. So there's a future testable prediction that once we do experiments, maybe mutagenesis on some of these differences in those proteins, we'll see, are they multifunctional or are they just reflection of descent with modification? So I'm just saying it's good you brought that up, and that just brings me to another prediction that creationists are making. But if you had another couple questions, yeah, go ahead and maybe we'll start wrapping it up there, Snake.
Yeah, so my first one was for synergistic epistasis, you basically say that it makes the problem worse for genetic entropy, but you also say it's invisible to selection. So I wanted to know how something can simultaneously be invisible to selection, but also make an organism extremely unfit. How can those things be happening at the same time?
So synergistic epistasis, for the audience sake, did you want to kind of explain like exactly what that mechanism is? What
Yeah,
it is
it's
proposed?
basically it's a process by which if mutations are accumulating, they interact with each other differently, and so the ones that interact with each other the worst basically make each other worse, and so it makes that organism or that particular child much more visible to selection. So if these mutations are interacting with each other, like say mutation A is bad, but if it's paired with mutation C, then it's really bad. So that will make mutation A get selected out of the population much more quickly. And the common response is that this accelerates genetic entropy instead of selects out the worst versions of mutation accumulation. But it's also simultaneously claimed, like by Jensen or Sanford, that it's also invisible to selection, even though if it's making the organism unfit, that's exactly how selection works.
Right. Okay. So good explanation. Yeah. More or less, if we have lots of these mutations, these nearly neutral ones that seem to be invisible to selection, because selection is going to amplify your best beneficial mutations. Of course, selection always gets rid of your most detrimental mutations. You have a mutation in one of those highly conserved mitochondrial DNA proteins, that organism's going to die. Of course, it's not going to live to reproduce. This is just basic. Everybody agrees with this. Now the question is, those nearly neutral mutations, right? Are they absolutely neutral? Well, they might be if the vast majority of our genome is junk. Or are they nearly neutral, as in they only have very subtle effects on organismal fitness? Are they just slightly deleterious? Well, synergistic epistasis, like you said, would suggest that if you have a lot of these nearly neutral mutations that selection can't see, once they accumulate enough, where now they can start interacting with each other and amplifying each other's deleterious effects. For example, the intensity of degeneration would be at a point where now selection can see the damage and get rid of that organism and apparently halt genetic degeneration. Is that more or less an accurate representation of what you're saying here before we move?
Well, alone it can't do it, but it's one of those processes which in combination with things like recombination will weed out basically the genetic dead ends, so to speak.
Right. So two points I would make. Obviously, like you said, you read those because I've often pointed to the numerical simulations that have been done that have been stated to falsify the synergistic epistasis mechanism. Actually, 3 points. Second point, somebody like Grar, what's funny, and he's the one that said if ENCODE is right, evolution is wrong. He's extremely against the notion that the majority of the genome is functional. And yet he's not even looking to synergistic epistasis. It's not this hero mechanism that all evolutionary biologists and population geneticists look to, because there's flaws in it. A lot of it is just it's more so it's a hypothetical mechanism. Can it do the job? Can it not? We may need more experiments. But here's why I say that mechanism fails to halt deleterious mutation accumulation. Well, the problem is when it comes to the human population, we've all been accumulating deleterious mutations. And for an evolutionist to say that they're not deleterious, even to a small effect, you'd have to actually prove that the majority of the genome is junk. But I actually find it quite easy to demonstrate that the majority is functional. I enjoy discussing those issues. So if all of these, all humans have been accumulating deleterious mutations, that means we're all multiply mutant. Maybe some a little more mutant than the others. All selection can do is choose the worst out of already a population of pretty degenerate humans, for example. I mean, no one's going to say humans are getting better. I mean, we see diseases are accumulating, for example. Here's the thing. So yeah, synergistic epistasis may theoretically be able to work in slowing down genetic degeneration, but when I say it just makes the problem worse. That's because whatever population may be experiencing synergistic epistasis, because those mutations that have been accumulating are now amplified to the point where selection's effective, that's just going to make that population go extinct quicker when the entire population as a whole is still slowly degenerating. I always make the point, you have a room full of people, everybody's equally mutant, some a little bit more mutant than the others. You can kill off the most mutant of people. Well, you still have now a room half full of people who are more mutant than the generation before, because we're all inheriting our ancestors' mutations, 100 new mutations per person per generation. So yeah, I would say, in a nutshell, synergistic epistasis, recombination, these mechanisms, yeah, it can slow down the degeneration process, but it can't actually stop or solve the problem. So I think that's where we would probably disagree. You seem to imply that it can actually solve the problem. I don't think it can solve the problem. Go ahead, Snake.
So, but are you saying that you accept that the ones in which synergistic epistasis is occurring, in which two mutations are making each other worse, that makes that individual more visible to selection?
I would say that individual, to the extent that synergistic epistasis is happening, it's happening so we know selection acts on the phenotype, not the genotype. So yeah, the selection has to see the individual, or for example, maybe a population that's highly degenerate, it has to see those effects. And if there's enough, let's say, nearly neutral mutations that have accumulated to the point where there is significant degeneration, the natural selection will get rid of that organism. It may help. It's a mechanism that can help slow down the degeneration process, but the degeneration itself is inevitable. We're still on a that mechanism is, I'm saying, it still fails to curb deleterious mutation accumulation. It might just result in populations of organisms experiencing rapid extinction a little bit quicker than they may have normally, because selection was able to see the amplified effect of all these deleterious mutations. Yeah, I agree, it's a real mutation. How much is it working? The experiments may not have been done to determine, but I would predict that to whatever extent it is working, it's only working to just slow it down even more, but the degeneration will happen. Go ahead, Snake.
Okay, so but the problems I have with Samford's model is that it straight up explicitly does not account for or include any beneficial mutations. It's zero. And so we have other simulations where Kimura showed that if we put even so synergistic epistasis alone, none of these processes, recombination alone will not stop mutation accumulation. Synergistic epistasis alone won't do it. None of these things alone, but when they're working together, Kimura has shown that, and when you include beneficial mutations, that the rate of beneficial evolution goes up super fast. And he was just doing beneficials at the same rate as deleterious, but they're actually occurring at like maybe a 1-to-3 ratio, and that model completely destroyed deleterious mutation accumulation. And we've also observed compensatory mutations, I guess, in nature and in the lab specifically, in real time, that have repaired deleterious mutations that occurred in the lab and no longer have that deleterious effect anymore due to these further mutations. And so when you actually include those in the model, Sanford's numbers don't make sense anymore.
Here's my objection to that is Sanford did include beneficial mutations while Kimura didn't. I think Kimura may have
no, I read the paper. It said I made a whole video on it. I show the part in the paper that says there are 0 beneficial mutations in Sanford's model.
Sanford explained, because even in the Lenski experiment, I think it shows that the beneficial mutations are so rare to the point where it may even have no effect on ultimate fitness. It's about like 1 in a million, and then even those beneficial mutations, there's evidence that they are reductive. So if they truly were the type of beneficial mutations that could counterbalance the net loss of genetic information, functional information, that is, then there may be a case there. But the thing is, these rare beneficial mutations that pop up are generally reductive in nature, or they're just epigenetic related, or general organismal adaptation, for example. I mean, that's not really going to do anything to curb the effects from deleterious mutation accumulation. But for example, like if you look at what I mean by reductive is, let's take sickle cell anemia, for example. People that have that mutation, okay, that cause that disease, they have a broken gene, which then results in a broken protein, okay? This broken hemoglobin protein then causes deformed red blood cells, and these deformed red blood cells will clot, which then leads to inadequate oxygen supply and leads to anemia. That's just it in a nutshell. But 2 doses of this gene, if you're heterozygous at that gene, you will die prematurely. Okay, this is deleterious, this is destructive, this is not an improvement. But why is it beneficial? Because all the evolutionists would point to that as, for example, a beneficial mutation. Well, for people who have that broken gene and broken protein, those red blood cells are defective to the point where malaria cannot thrive in the person with that condition. Therefore, those people are more resistant to malaria. The sickness, though, associated with the disease results in a resistance to the malaria parasite. Overall, it's reductive. It's not taking things forward. And that type of beneficial mutation, which is the majority of them, for example, are not going to counterbalance the damage. So Kimura, if he's making, if evolutionists are trying to curb the deleterious mutation accumulation by adding in beneficial mutations. They're too rare. And when they do happen, they're reductive in nature and they are functionally compromising to the genome. So that would be my point. Go ahead, Snake.
But it's not zero. Like I can show you the screenshot of Sanford's paper saying beneficials equals none. So he's saying it's absolute zero. But we've observed in the lab that beneficials do in fact occur. And I'll find the paper also. I think Sanford actually cites this, which says that beneficials occur at about a rate of 1 to 3 beneficial to deleterious. So that's basically the problem there. But I can send that to you after the show.
I'm pretty sure I'll have to look it up again. I've read genetic entropy many times, but it's actually been a while since I've read it again, since there's so many other papers and new books that I've been reading. But I'm almost certain that he agrees beneficials do happen. They're rare and they're generally reductive. Therefore, adding them in or not adding them in, it doesn't matter because it doesn't change the curve, because deleterious mutations are accumulating. The question is, can beneficial mutations that even evolutionists will agree are rare, can they actually counterbalance the damage? And he goes into like Lenski's experiment, which shows that beneficials, for the most part, are rare to the point where they only happen in 1 in a million mutations. And even then, there's evidence that they're reductive. So the question is, are they even going to counterbalance the damage? But we've been going on for a while, and we got to get our next stream in with Jeremy. I know you guys are probably just going to talk evolution. You don't have to do astronomy, but since you're my guest, Snake, and it's been a really good discussion. It's been respectful. I hope you felt you've had enough time to talk. I'm going to give you the last word. So make your final points. Give me your final thoughts as to how you think the conversation went. Anything you want to address that you feel like you didn't. Go ahead. You get the last word.
Yeah, I think I had 2 points that I didn't quite get to. So we actually made pretty good headway, I guess. So the one that I was going to bring up that I hoped we would get to was the DNA barcoding of the CO1 gene. And I mean, it has a relevance to a wider point as well, too. Where in your video, you were talking about how the canine kind basically could be grouped into a kind because of the similarity of the DNA barcoding of this one gene, and the feline kind as well. And the range of difference in them was, so for the canines, it was 80 to 108 base pair difference, and with the feline kind, it was 82 base pair difference. And then, so that was defined as a kind, but then with the humans and the chimps, there was a 65 base pair difference. So that would be within the range of a kind still. And that's to connect that to the larger point is, for example, there's a larger difference between chimps and gorillas than between chimps and humans. And there's a larger difference between the house cat and the lion between chimps and humans and things like that. So, I mean,
well, real quick,
we could talk about.
I know I said I give you the last
for a whole another 3 hours.
Yeah, well, because I meant last word is in like wrap things up, and now you brought up a new topic with the barcoding. But I will say real quick, I still want you to have the last word, but let me just address that. Then whatever you say next, just make sure whatever your last word is, maybe like a rebuttal to something that we've already talked about, because the DNA barcoding, the CO1 gene, also highly conserved mitochondrial DNA protein. Yeah, I said it a few times in this debate. Yeah, though that CO1 gene, the differences also reflect nested hierarchical patterns. The big thing, so that's why the function and the molecular clocks are still your two main lines of evidence, and of course the testable predictions that flow from the data. But those are the two lines of evidence that are going to be able to differentiate between universal ancestry or limited ancestry. Now, the point with the DNA barcoding in the CO1 gene is that the diversity in that gene is limited, and that's where that paper comes into play, the new one that suggests that over 90% of the species in the world today came about at exactly the same time. Like there was one big worldwide genetic bottleneck of all of life. So that is good supporting evidence that there's been a bottleneck at some point in the past. For example, according to our model, that would be the, as you know, the Ark, Noah's flood. So that genetic boundaries study does, like I said, indicate that I think the evolutionists, to explain those results, the authors of that study, they speculated that life got nearly wiped out across the board. They say about 100,000 to 200,000 years ago, and then had to restart. The dates, that's just the calibration of the fossil record. Again, that's not based on the genetics. But it's funny because, yeah, there was a nearly extinction event. It's called the global flood. So that's the bigger picture of the DNA barcoding. But I do agree that whatever gene you look at, they all follow a hierarchical pattern that seems to reflect descent, but it also reflects design. The question is the differentiating evidence, the DNA function, the molecular class. So I agree with the hierarchies. We can get into the specifics of like the felid kind, for example, and whether or not God created animals. Because remember, God created two people, Adam and Eve. So we would expect low genetic diversity. That's what we see today. God created animal kinds in bulk, in populations. Therefore, we would expect a higher genetic diversity. It appears God created animal kinds with a greater ability to adapt, to diversify, for example, because we see more DNA differences in the nuclear DNA and species of the same kind than we do between, say, humans and chimps. So why don't creationists say humans and chimps are related? Well, that's because patterns are agnostic. Number of DNA differences are agnostic. It comes down to molecular clocks, DNA function. I said that many times. So I hope that kind of answers your question with DNA barcoding. But you're right. We could go on and on for that for 3 hours. Because I did address the barcoding, the CO1 gene, in your last words, if you want, you can address what I just said, wrap everything up, and boom, we'll be good to go. Go ahead, Snake.
Yeah, I still don't see like a consistent pattern there, but I'll circle back to that. The only thing that we didn't get to that I wanted to get to, which I think we should talk about next time with the mitochondrial mutation rates, I was just going to bring up the Drosophila orphan genes, where we can genetically show that these species are the same genus and they're related, and their orphan genes are being born and dying off. So they're not lineage specific. So yeah, we'll save that for next time. But as for, I guess I don't want to repeat myself too much, but basically I just don't see a consistent enough pattern of design. So what would like the main points for going back shortly to the CO1 gene? There should be a pattern of similar sizes for each, similar difference range for each kind, and we don't see that. We see chimps and humans should be the same kind by that same the same standard that we use for other kinds, and I don't see consistency there. If we're talking about the cytochrome C gene, we're still seeing only consistent patterns that fit evolutionary hierarchies and not design hierarchies, since there's really no reason that worms would need to have a sequence difference that was so much different than the chimps and the humans. If the chimps and the humans were evolving at the same time for the same amount of time, then we should see the same amount of differences between each kind. And since, I mean, even Linnaeus put humans in the ape category because morphologically, anatomically, genetically, they're the most similar, and there are very few differences between them. And by the same standards, the same patterns that we use to distinguish other kinds, if we're trying to be consistent, we can't separate the humans from the apes. And patterns, embryological patterns, there's no reason for a designer to make things just to take it off. And that's not even a scaffolding problem. Like the way feathers grow, they grow in a cone, and basically most of that cone is scaffolding, and so a lot of the scaffolding dies and falls off, and there's a branching pattern of keratin between that. But that's not what we see in the embryology of things like whales, where the legs grow and they're not scaffolding for anything. They just grow and then they die off. And that's not a feature that we find in any designed anything ever. And I guess going back to the beginning with the inconsistent design patterns, if we're going to have a swimming animal that lives in the ocean, then it should be designed with gills. Now, it makes more sense and fits the pattern that if it was on land and it came into the ocean, like an otter from a weasel, well, the otter's not going to all of a sudden just sprout gills. It's going to have to derive those gills from some sort of tissue that absorbs oxygen and water, and that's not going to happen in very short order. So, and it's not going to happen to a whale either. But if gills were there first, then it makes sense that they can be lost. And if air bladders were there first, it makes sense that they can gain a better oxygen absorbance. And again, that pattern, we don't see a pattern of design things where we have fish that have air bladders and lungs. We see fish that have gills and lungs, but we see fish that have gills and air bladders, but we'd never see fish that have gills, air bladders, and lungs at the same time, which is something that God can do and easily could do. Same with the gills on whales. That would be a pretty good evolution destroyer. And if God wanted us to believe in creationism, he could throw in roadblocks like that pretty easily with his foreknowledge and foresight. He could completely destroy a lot of the evolutionist arguments just by creating a lot of creatures that disrupt this pattern completely. Were there any more examples of those were just a few examples of the patterns. I just don't see it fitting a design pattern, and every time, even you're saying that it's fitting a nested hierarchy, and we're able to predict without even knowing like which hierarchy is closer to which, and we're able to predict what hierarchy a fossil is going to belong to before we even see it. So that's basically, it's really hard to point out patterns just with words. Even with pictures, it's hard, but that's basically the summation, I guess, of what I was trying to bring up tonight.
Well, that was a long closing, Snake, but I did promise you that I'd give you the last word, and it's been a good discussion. I hope the audience had fun. I think we touched on a lot of good topics, some topics that are important to discuss and probably need more discussing. So we're going to end it here because it looks like Jeremy's in the chat too. He's eager to have his round. We're going to keep it the same, respectful. Just a nice free-flowing dialogue. So this was good. Me and Snake have done this many times before, and I think this has probably been my favorite one. So thanks again, Snake. You're free to take the video and upload it to your channel as well. But yeah, I'm going to shoot it over to Brother Matt to shut it down. And I think we already have the next stream ready and good to go. So we'll shoot out links in 5 minutes. Okay.
All right.
All good, Matt.
Okay.